Every aquarist knows this. The blue-green film, the smell, the faint sense of personal failure. Most of us respond by removing it, adjusting flow, checking nutrients, and hoping it doesn't come back. Occasionally wondering what exactly it is. It is the oldest photosynthesizer on Earth. It oxygenated this planet's atmosphere. It is still, by most measures, winning. And this lab has been studying it for years. What the lab found about cyanobacteria changes how you read the thing on your glass. Some aquarists call it blue-green algae. This is incorrect in 2 of the 3 words. It is not always blue-green. The pigment ratio varies by strain and can produce greens, browns, reds, and near blacks. And it is also not algae. Algae are eukaryotes. Cyanobacteria are bacteria. There is no nucleus. There are no chloroplasts. No membrane-bound organelles of any kind. The photosynthetic machinery, aka the thylakoid membranes, the electron transport chains, they're embedded directly in the cytoplasm, running without the cellular infrastructure every other photosynthesizer on this planet takes for granted. In the filamentous strains, some cells differentiate into heterocysts, which are enlarged cells that perform nitrogen fixation under conditions of nitrogen scarcity. One cell in the chain doing a completely different job from its neighbors. Division of labor in a prokaryote, without a nervous system, without a brain or anything else resembling a decision-making apparatus. Just chemistry responding to chemistry. The blue-green film on your glass is a prokaryote. It shares more cellular architecture with bacteria causing your fish's fin rot than it does the algae growing next to it on the glass. They've been operating this way, unchanged, in fundamental design for approximately 2.7 billion years. The color variation in this culture collection is itself an argument. The name blue-green algae was coined by people looking at one strain in one context. The actual pigment palette of the cyanobacteria spans the visible spectrum, depending on which combination of chlorophyll, phycocyanin, and phycoerythrin the organism is expressing. The name, like most common names in biology, is useful fiction. Approximately 2.7 billion years ago, cyanobacteria evolved oxygenic photosynthesis. This was not a small development. This was, by any reasonable measure, the most consequential biological event in Earth's history. And cyanobacteria did it with no planning, no intention, and no awareness of what they were about to do to the planet. The byproduct of oxygenic photosynthesis is oxygen. Before cyanobacteria, Earth's atmosphere contained almost none. The oceans were full of dissolved iron. The land was bare. The existing microbial life was anaerobic. Oxygen, for them, was a poison. Over hundreds of millions of years, cyanobacteria filled the oceans, the atmosphere, and in doing so, triggered the Great Oxygenation Event, a global chemical transformation that oxidized the dissolved iron in the oceans, forming banding iron formations that are now mined for steel. And they raised the atmospheric oxygen levels that made aerobic respiration possible. They also caused, almost incidentally, the first mass extinction in Earth's history. The anaerobic organisms that had ruled the planet were poisoned by the atmosphere cyanobacteria produced. Every organism in this tank that breathes oxygen, the fish, the shrimp, the snails, the aerobic bacteria, and the substrate, they are alive because cyanobacteria spent 2 billion years poisoning the planet with a gas that now sustains it. The debt is not metaphorical, it is biochemical. The lab flask is not a curiosity, it is a living archive. What is photosynthesizing inside that glass has been photosynthesizing by the same mechanism since before the first animal existed. Cyanobacteria move. Not quickly, not dramatically, but deliberately and for a single-celled organism with no nervous system with remarkable precision. The mechanism is Phototaxis, a directional movement in response to light. Cyanobacteria detect the intensity and direction of incoming light using photoreceptor proteins embedded in the cell membrane. They coordinate a gliding motility using structures called type 4 pili, essentially grappling hooks at the molecular scale to move towards a favorable light source. There's no brain organizing this. There is a receptor, a signal cascade, a behavioral output, the complete circuit of stimulus and response running in a cell 1 micrometer across. The accumulation you see is not a metaphor for intelligence, it is signal transduction. Stimulus detected, signal propagated, motor behavior adjusted. The same fundamental logic that runs in every neuron in your brain simplified down to its irreducible elements and operating without a neuron in sight. The blue-green film that migrates across your glass overnight that accumulates on whatever surfaces are in the light that seem to always find the brightest spot in your tank are doing exactly this. The experiment you just watched in a petri dish is the explanation for the pattern you have been scraping off your glass for months. Not all light is equal to cyanobacteria. The photoreceptors that drive the phototaxis are tuned to specific wavelengths, and the response varies accordingly. Under red light, and the wavelengths that drive photosystems 1 and 2 most efficiently, the phototaxis response is strong. The organism moves towards it with purpose. Green light tells a different story. Chlorophyll A absorbs red and blue efficiently, but reflects green, which is why plants are green in the first place. Green light is, from a photosynthetic standpoint, the least useful wavelength in the visible spectrum. The phototaxis response under green light is weaker, or in some strains absent, or even reversed. What this reveals is not just a behavioral quirk, it is a window into the organism's internal model of the environment. Cyanobacteria are not simply moving towards brightness, they are moving toward photosynthetically useful brightness. The photoreceptors are doing quality control on the light, filtering for what the cell can actually use. The Aquarium LED over your tank produces a full spectrum output, including the red wavelengths that cyanobacteria find most useful. If the conditions in your tank are right, low flow, excess nutrients, low competition from other photosynthesizers, cyanobacteria will find the light, read it as favorable, and move toward it. Not because they are a nuisance, because they are extraordinarily good at being cyanobacteria. Keeping cyanobacteria alive in a laboratory is, in some ways, straightforward. They are not fragile, they are not particular. What they require is light, water, dissolved carbon, and a source of nutrients, specifically phosphorus and nitrogen, in their most accessible forms. Given those inputs, they grow. What is more revealing is what they do not require. They do not require oxygen. Some strains are facultatively anaerobic. They do not require complex organic compounds that most organisms need as building blocks. They are primary producers in the most fundamental sense. They build organic matter from light and inorganic inputs, and they do it with a metabolic flexibility that most organisms cannot approach. Limit the light, and they slow. Limit the nutrients, and they slow. Give them both in abundance in still water at a warm temperature, and they bloom. The lab is not performing a miracle. The lab is providing what cyanobacteria have been finding in ponds and lakes and slow rivers for 2 billion years. The low-flow corner of your tank, where the blue-green film keeps appearing, is in some ways a culture flask. Warm, still, nutrient-rich, plenty of light. The cyanobacteria found it because it offers exactly what the lab provides intentionally. Your tank is not failing, it is succeeding at creating cyanobacterial habitat in a specific location for a specific reason that your probes can identify. What does cyanobacteria in your tank actually mean? Not as a problem to solve, but as a signal to read. Location is the first variable. Cyanobacteria prefer still water. If the bloom is localized to a low-flow corner, under a decoration, on the light-facing surface of the substrate, the tank is telling you that water movement in that zone is insufficient to disrupt the surface colonization. The fix is not scraping, the fix is flow. Nutrient loading is the second variable. Cyanobacteria are not strong competitors in a tank with healthy plant growth and controlled nutrient levels. They appear when the nutrient balance tips, when the phosphorus or the nitrogen exceeds what the plants and algae can consume, when the decomposer loop is processing more organic matter than the tank's biology can handle. The cyanobacteria are the readout. The nutrients are the story. Competition is the third variable. In our previous episode, we established that green spot algae, a charophyte, is a sign of healthy competition. Cyanobacteria, by contrast, appear most aggressively where competition is the weakest. New tanks, sparse plantings, perhaps a recently disturbed substrate. They are not invading a healthy ecosystem, they are colonizing a vacancy. In every episode on this channel, we have argued the same thing. Every organism is a data point. Cyanobacteria in your tank are not evidence that you have failed. They are evidence that a very successful prokaryote has found in a very specific corner of your aquarium the conditions it has been exploiting since before there was oxygen in the atmosphere. The question it is asking you is, what created those conditions, and do you want to change them? This flask contains the oldest photosynthesizer on Earth. It oxygenated this planet. It drove the first mass extinction. It gave rise, through endosymbiosis, to every chloroplast and every plant you have ever seen. It is still, by any measure of evolutionary success, one of the most accomplished organisms in the history of life. It has survived 5 mass extinctions. It thrives in hot springs, in Antarctic ice, in the open ocean, and in the low-flow corner of a freshwater aquarium in a building it has no awareness of on a planet it has not finished reshaping. The thing on your glass has been doing this for 2.7 billion years. It found a warm, still, nutrient-rich surface. It moved towards the light, as it always does. It built a mat, as it always does. It is not a pest that arrived in your tank. It is a survivor that found in your tank the same conditions it has been exploiting since before the first fish existed. That does not mean you have to let it stay, but it does mean you should respect what you are looking at.